pH. Finally, each enzyme shows its highest activity at a particular temperature, pH, called optimum temperature or optimum pH. Now finally, activity declines both below and above the optimum value. Okay. Now, if you carefully observe, low temperature, what is that? Low temperature preserves the enzyme in a temporarily inactive state, where high temperature, what is that? High temperature, okay, inactive state where high temperature generally destroys enzymatic activity, because proteins, whatever they are here, are denatured by the heat. Concentration of the substrate with increase in substrate concentration, the velocity, what is that? The velocity of the enzyme reaction rises faster. Now reaction ultimately reaches maximum velocity, what is that? Maximum velocity, okay, which is not exceeded by any further rise in concentration of the substrate. This is because enzyme, whatever it is there, having molecules, okay, are very fewer than that of substrate molecules, and after saturation of these molecules there are no free enzymes. After saturation, free enzymes will be not there, okay, everybody will be busy with their work. So no free enzyme molecules to bind with the additional substrate, so the substrate will remain like that for some time. The activity of an enzyme is also sensitive to presence of specific, what is that? Presence of specific chemicals. Presence of specific chemicals that can bind to the enzyme. Now, when the binding of these chemicals, whatever there occurs, you can see in the diagram what happens. Okay. In this figure, you can see the pH balance. Okay. On one axis pH and another axis you can see the enzyme activity. So, and temperature and energy activity, you can see in the B diagram how the curve is looking like. Concentration of the substrate, here you can see velocity of the velocity reaction V max, and substrate S. You can see a kind of curve which is obtained here. Understood? So already we know all these things. Shut off enzyme activity, the process is called as a inhibition. Inhibition is nothing but shutting off or shutdown of the activity of enzyme. When the inhibitor closes, inhibitor is a structure which can close the closely resemble the substrate in its molecular substrate. Inhibits the activity of enzyme. Inhibitor is a structure that can stop the functioning of an enzyme. It is known as competitive inhibitor. What is that called? Competitive inhibitor. Due to its close structural similarity, with the substrate, the inhibitor competes with the substrate for the substrate binding site of enzyme. Consequently, substrate, if you carefully observe, the substrate cannot bind as a result. Now, the enzyme action declines. Inhibition of the example, let us take example for this. Inhibition of the succinic dehydrogenase by maltose. Succinic dehydrogenase is an enzyme. Its inhibition is occurring by the malonate. What is that? Malonate. It is malonate, which closely resembles substrate succinate in structure. Finally, such competitive inhibitors are often used in the control of bacterial pathogens in our body. Classification and nomenclature of enzymes will continue in